Tuberculosis, or otherwise known as TB or MTB, is a potentially serious and even fatal infectious disease caused by Mycobacterium tuberculosis that mainly affects the lungs, but in rare cases can also enter the bloodstream and get to other parts of the body, such as the brain, bones, kidneys, and other organs. MTB is a rod-shaped bacteria that is surrounded by a mycolic acid structure, which adds to its virulence factors and is responsible for developing resistance to certain antibiotics and lysosomal components of phagocytes. Tuberculosis has a remarkably slow growth rate, which may not elicit a sufficient response from the immune system to eliminate it. When detected, MTB elicits a severe inflammatory response in the form of granulomas, which contain a collection of immune cells and macrophages. Granulomas form when the immune system attempts to wall off substances it perceives as foreign, but are unable to eliminate them. In many cases, MTB causes such a large inflammatory response, these granulomas break open and turn necrotic, which can lead to tissue damage and symptoms such as fever, cough, bloody mucus, and weight loss. This form of tuberculosis is known as TB disease or active TB, which can occur in the first few weeks after infection with TB bacteria, or it may occur years later. If the granulomas heal before being broke open, tuberculosis bacteria remains dormant within these healed granulomas in a dormant state which is known as latent tuberculosis. Individuals who have latent tuberculosis show no signs and many times are unaware that they have the disease until these bacteria are reactivated at aerobic sites later in life. Tuberculosis is spread through the air when someone with active TB of the lungs or throat coughs, sneezes, speaks, or even sings, and people nearby breathe in these harmful bacteria.
Once a person has breathed in droplets that contain TB bacteria, they travel down the trachea and enter the lungs, where they collect in the alveolar sacs. Once in the alveolar sacs, the bacteria begin to multiply. The body's immune system starts working and macrophages begin to surround the bacteria. A granuloma is formed to keep the bacteria from spreading. The TB bacteria remain in the lungs, but the body is protected from disease by the granuloma. In 8 to 10 weeks, the person will most likely test positive for latent TB infection. The progression from latent TB infection to TB disease occurs when the granuloma break open and the TB bacteria multiply. Then the person becomes sick with TB disease and may be infectious. This progression can occur immediately after infection, many years later, or not at all.
Tuberculosis bacteria is responsible for suppressing the immune system by 3 specific mechanisms. First, MTB is responsible for the inhibition of phagosome maturation by inhibiting phagosome acidification. Under normal conditions, the VATPase pump pumps the protons into the phagosome to increase its acidity, which aids in the maturation of the phagosome. The mycobacterium releases a protein within the phagosome called PTPA protein, which binds to a subunit of ATPase that is responsible for the inhibition of the pump, which results in no acidification of the phagosome and ultimately no maturation. Secondly, inhibition of phagolysosome formation is another way where MTB is able to suppress the immune system. In this process, the phagosome and lysosome within the macrophage are unable to fuse together, which ultimately leads to bacteria surviving inside the phagosome. Normally, the phagosome has a Rab5G protein on its outer membrane, which can be exchanged for a Rab7G protein, marking the phagosome for fusion with the lysosome. But the mycobacterium that is encapsulated within the phagosome recruits a Rab22AG protein, which prevents the exchange between the Rab5G protein and the Rab7 protein, ultimately leading to no phagolysosome fusion and no elimination of bacteria. The third and final way that TB is responsible for suppressing the immune system is by the inhibition of interferon production. Within a macrophage, there are C-gas molecules that are responsible for the detection of foreign DNA and activating the STING, or otherwise known as stimulator of interferon genes pathway, which ensures the production of interferons by the macrophage. Since there is no fusion of phagosome and lysosome, the foreign DNA cannot be degraded and exposed to C-gas, which leads to no detection and no STING pathway activation. When the STING pathway is unable to be activated, there is no interferon production. One of the most widely used drugs for the treatment of TB is isoniazid, which is a prodrug that needs to be activated by the bacterial catalase peroxidase enzymes produced by Mycobacterium tuberculosis. This is known as CAT-G. The mechanism of action for isoniazid includes, first, that CAT-G needs to cause the formation of isonicotinic acyl radical. This radical couples then with an NADH to form nicotinoyl NAD adduct. The complex then binds to a carrier protein, reductase, resulting in blockage of the natural substrate and action of fatty acid synthase, which ultimately leads to the inhibitionist synthesis of the mycolic acid structure, which is responsible for killing rapidly dividing mycobacterium within the body. The green structures display isoniazid bound within the catalase peroxidase enzyme, or otherwise known as CAT-G enzyme of Mycobacterium tuberculosis, where it inhibits or eliminates the growth of mycolic acid on these bacteria. Tuberculosis is a serious infectious disease that can potentially wipe out large amounts of people in a population if left untreated. Thankfully, due to the direction of positive research, diagnosis, and treatment, TB is relatively unheard of in the U.S., but it is important to continue to educate the public about the disease to encourage those who are undiagnosed or untreated to seek medical attention immediately.